Thank you. We are switching gears today simply because you're starting to hear about all these wonderful optical technologies, but we want to know why and how can we apply those. And of course, the main focus of most of these optical technologies is the imaging and sometimes even the treatment of certain diseases. Specifically, faculty here are interested in cancer. So I don't know how many of you had any biology during college and when was that? So just to remind you a little bit about what are we trying to image and what are the properties of some of these diseases. So if you type nanobiophotonics, and of course some time ago when I started to be involved in this effort in Beckman Institute, I didn't know what nanobiophotonics is, so I just used the Wikipedia to type in nanobiophotonics, and this is what came up. Not actually it's a joke, but the description. It's observing very small live structures using light. And we are, of course, interested in observing or imaging live structures. So what is the smallest live structure? It's very hard to define life. If you think about it, how can you define life? There is no definition. So scientists actually defined a certain properties of life, of living structures. So they know what's living and what's not living. So, and these are the 7 basic properties of any living structure or organism. So living things are highly organized. They're homeostatic, they have ability to reproduce themselves, they grow and develop, respond to stimuli, they're adaptable, and they can take energy from the surrounding, from the environment. They're actually thermodynamically open system. They take the energy, they process the energy, and they use it for their own processes. So what is the smallest structure that we call life? That's the cell. Cell definitely is not the smallest or the simplest structure that we know. Every cell is actually containing a lot of very complex structures that we call organelles. Organelles are made of macromolecules and macromolecules of simple molecules, and we can go on to subatomic particles. But what's interesting is no matter how complex the structures below the level of cells are, they're not considered live because they don't have all these 7 properties of living organisms. So if you take a mitochondria or a nucleus out of the cell, it's not considered a living organism. Cell is, because it contains all these 7 properties of life. Cells, of course, are, as I said, they're considered the smallest unit of life. But they're organized into tissues. Tissues are organized into organs, organs into organ systems, and organ systems into a full organism. But what's very important to remember, if you're ever studying any live structures, is whatever properties you have at one level, the next level contains the properties that are not only the sum of the levels below, but the new properties as well. For example, a tissue. If you take any tissue, like epithelial tissue, it has all the properties of epithelial cells. But it's not just a group, a bunch of epithelial cells. Those are the cells that are highly organized and communicate between themselves to give more properties at the level of tissue. So very often, especially in the past, when cell biologists were exploring the cells, they would say, oh, epithelial cells have these properties, so does the epithelial tissue. And then they start ignoring very important things, like communication between the cells, signaling, ECM, we'll see that's extracellular matrix, extracellular environment, and so on. So keep that in mind if you are ever studying cells. We don't have time, but just to remind you how complex each cell is. This is a general structure of an animal cell. Why I emphasize animal? Because if you ever work with plants, you'll see that there are some basic differences between plant and animal cells. But what's characteristic for every cell is that it's surrounded by the cell membrane. And cell membrane is not just a membrane that prevents the diffusion, the free diffusion between intra and extracellular environment, but also has a number of very important functions, specifically communication between the outside environment and inside the cell. And of course, within the cell, we have a lot of different organelles or structures that have very specific structure and function. And they're immersed into the cytoplasm or intracellular environment that also has actually very specific structure. Now we realize that everything that's inside a cell has a very specific structure and function. And this is the membrane that is, as I said, the barrier for free diffusion between inside and outside the cell. But again, it has a very specific structure and very specific function. And you can see it's very tightly connected physically and functionally with the extracellular environment that we call ECM, or extracellular matrix. So there are a lot of proteins, polysaccharides, and they're all very tightly connected. And of course, even now when you start talking about different pathological conditions, including the cancer, all these parts inside and outside the cell have a very important role in those diseases. So we cannot ignore them. So this is just one typical immune cell that belongs to the immune system. In seeds has a very specific structure, and this is actually a lymphocyte that's going around and eating debris and viruses around your body and keeping your body clean and healthy. And this is important to remember, that cell is a living unit that's greater than the sum of its parts. Now what I want to talk about is to move on to one of those properties that define life, and that's the ability of cells to reproduce, okay, to divide and give more new cells. So what is the cell reproduction or division important? It's important for reproduction. What kind of reproduction? If the organism is only one cell, and there are organisms that are only made of one cell, division actually gives not only 2 new cells, but it gives 2 new organisms. So that's one of the purposes of cell division. But in multicellular organisms like us, the cell division is important for growth and development. You all know that we start from one cell, and when the cells start dividing quickly, we grow and differentiate and develop in a full grown organism. And then, when you reach the full size, let's put it this way, then you still need the cell division for tissue renewal, because some of your cells actually get damaged or wear out and die, or they're killed or injured, and they need to be replaced. So even if you're a fully grown adult, you need a cell division for tissue renewal. Now, different tissues, different types of cells have different capacity for cell reproduction, depending on their function and the level of differentiation. These are different types of epithelial tissues. Where do we find epithelial tissues? We found them on all surfaces, right? Outside and inside surfaces. They actually protect organs. They protect the skin, of course, but not only skin. All our cavities, all organs are covered with epithelial tissue. So thinking about it, you can very quickly conclude that these epithelial cells are exposed to all kinds of abrasions, chemical, mechanical, and they're easily injured, damaged, and they very often need to be replaced. And that's why epithelial cells have a very high capacity, ability to divide. And we'll see why is that important for cancer. Some other cells, like muscle cells or nervous neurons, actually not only they have a low capacity, they lose capacity to divide. Fully differentiated muscle cell or neuron, which is the basic cell of the nervous tissue, completely lose the capacity to divide because they differentiate so much. They lose that ability to divide. And that, of course, as you know, probably can cause a lot of problems if you have an injury in your spine and there is some cut in neurons, especially the axons. They very often don't have ability to regenerate because they don't have ability to produce new neurons. So it depends on the type of the cell and the level of differentiation. Now, we need to talk a little bit, I'll go quickly over this, but in order to understand where the cancer as one of the biggest threats of our civilization is coming from, we need to just touch upon the basic cell division that we call mitosis. Okay, so the basic cell division is called mitosis. Now, you know that in the cell, we have a nucleus, and in the nucleus, we have a DNA. That's the basic genetic material. We don't see that genetic material with just light microscope under normal circumstances. But when the cell decides to divide, that DNA material starts condensing and supercoiling into very specific structures that we call chromosomes. And we can see them. And actually, people first discovered chromosomes because they're visible with the light microscope. And then they didn't have a clue what they do and what they're made of. And then later on, they discovered actually that they're made of DNA and that is the main genetic material. But these super tight and super coiled structures are visible with the light microscope. And they have a very characteristic structure. And of course, every in each cell has a very defined and specific number of chromosomes. Chromosomes that depend on a species. So how many chromosomes do we have? 46, 23 pairs exactly, and this is how many DNA molecules are in our cells. But other species can have from 1 to hundreds. Especially some plants, it's interesting, that doesn't give them too much advantage, but they have up to 100 different DNA molecules or chromosomes. So every cell is going during its normal lifetime through what we call the cell cycles, through certain phases that are repeating themselves. So the first phase is called G1 or gap 1 phase. This is just the phase where cells do what they're supposed to do. They're functioning normally, doing their job, until at a certain point they decide, and we'll see what does it mean decide, that they receive a signal from inside or outside the cell that they need to divide. And somewhere here we'll see there is a point, a decision point. Then the cell enters the next phase that's called the S phase. It stands from synthesis, and this is the period where the DNA is replicated, because if you want to divide one cell into two, each cell needs to get exactly the same genetic material. So the first essential step in cell division, when you don't see any other changes, is the replication of DNA. And that's happening in this S phase. Now in the next G2 phase or gap 2 phase, what cell is doing is just preparing, making a lot of different proteins, making mitotic spindle, a lot of different organelles and components that are important for the cell division. And then we have, actually you can see that this phase is very short compared with the complete cell cycle. There is that mitosis. There is that phase where the actual replication or division of the cell occurs. And it's a continuous process, but we always like to cut things into pieces so we can analyze it better into different phases. And the cell division, these are the actual images, if you want, where a nucleus and the chromosomes and the cytoplasm are nicely colored with different stains so we can see them better. And those are, of course, the diagrams representing what's happening during each phase. So pretty much without any details, first we have the formation of chromosomes. Why is that important? Well, if you want to divide genetic material into two absolutely equal, identical parts. You need to manipulate them, right? So in order to manipulate those DNA molecules, they need to be really tight and nice little structures so you can divide them into two. So the first phase is coiling of DNA molecules into chromosomes, and you can see the formation of very important component part of the cell during the cell division, and that's called mitotic spindle that's made out of microtubules. And this structure here is actually going to facilitate the division of chromosomes into equal parts, into equal components. So in the next, and if you had biology in high school, maybe you remember that this is the prophase and this is the pro-metaphase. You see that these chromosomes that are already divided, duplicated. So each of these chromosomes actually contains two identical DNA molecules that still stick together. They are actually attaching to the mitotic spindle and waiting to be divided. So during the metaphase, they're nicely lined up. In the anaphase, they divide, they separate, and move toward the opposite sides of the cell. And as the final, actually less important phase, we have the cytokinesis, or the division of the cytoplasm itself. So during the cell division, keep in mind that dividing the cytoplasm and organelles, it's not important. The only thing that's important is how you divide the genetic material. And a lot of genetic problems and, abnormalities are stemming out of the fact that maybe genetic material either didn't replicate correctly or didn't divide between the cells correctly. Okay. As I mentioned, the frequency of these cell divisions varies from one cell type to another, and it's tightly regulated at the molecular level. Cell needs to divide when there is a need, but it shouldn't divide if there is not a need. So you can imagine that there are a lot of signaling and control pathways that are regulating this cell cycle. And we will see that there are different points during the cell cycle that are in charge of controlling this. And of course, we won't have time to discuss all of those. The most important, checkpoint, let's put it this way, checkpoint is right here, I told you, in G1 phase or gap 1 phase. That's called restriction point, usually, restriction point. Because cell doesn't want to invest energy and time into replicating DNA and dividing and going into the same process. So the G1 is the phase where it's most efficient to make this decision whether to divide or not divide. And you can see that we can actually represent this cell cycle in the form of a clock. But there are also two other checkpoints in the G2 phase and M phase. If something goes wrong, why is this important? Why is this checkpoint important? Well, if there is a need for a cell to divide, but there is a mistake in how the chromosomes are separating. Maybe, let's say we have 46 chromosomes, and one side of the cell is getting, 44, and the other 42. Well, that's not good, right? So this is the checkpoint where cell checks, literally checks the number of chromosomes that are migrating toward the opposite poles. And if there is a mistake, there is a process in place that will stop the further mitosis and literally disintegrate the cell. How is it possible? Did you hear about apoptosis or the cell suicide? So some of you did. There is a very complex process that we didn't know about until about, 10 years ago maybe? We thought always that the cells die accidentally or something kills them. Well, that's not true. Cell has what we call the programmed suicide. It's called apoptosis. If anything goes wrong with the cell, including during the cell division, it starts the process of eliminating itself so it doesn't hurt the other cells and the other parts of the body. So that's called apoptosis, or the programmed suicide, controlled suicide. Okay, so this is the checkpoint I told you about. That's called the restriction point. And this is the point where a cell just looks around and says whether it's really necessary to go into division or not. Now, in the case of epithelial cells, I told you very often, there is a need for cells to replicate, so they'll pass this checkpoint and continue with the cell division. Very often, once you reach the full size, the full growth, your cells, like muscle cells, neurons, liver cells, they just check out, okay? They leave the cell cycle and go into what we call G0 phase. That means they're just dormant. But a lot of cells have ability to get back from that G0 phase. For example, you all heard that you can transplant, if somebody needs a liver transplant, you can take a piece of liver from a live donor and put into the patient, and both individuals can grow liver to the full size. Liver has a great ability for reproduction and when it's needed to get to the full size, the full growth. However, muscle cells and neurons don't have this ability. Once they check out of the cell cycle, they can never come back. And that's a potential problem. Okay, I'd rather skip this one. Just to tell you that there are molecular mechanisms. Specifically, there is cyclin. So, you know, if you ever work with these mechanisms, that rings the bell. There is the cyclin, which is a protein, and the enzyme cyclin-dependent kinases that are actually as a complex activating the cell division. So if the levels of cyclins are increasing, they're forming the complex with the enzyme and starting the process of cell division. If the cyclin levels are low, then there is no complex and cell division does not go on. What is controlling the growth of our body, which means the cell reproduction, basically? Those are those different proteins like cyclins, different growth factors, growth inhibitors, because there is always a balance between the growth stimulators and inhibitors. There are some suppressor genes and oncogenes that we are going to mention in a minute. Now, what is so the signal is at the molecular level. Certain proteins, certain signaling pathways, but what is activating those signals? A lot of external and internal factors are important for starting those signals. Now, let's talk a little bit about two main external factors. Why would cell go into cell division? Well, cell doesn't know, yes, you need to divide because a certain number of epithelial cells died. What does cell know? Well, it can sense changes in the environment. How do we know this? From experiments, of course. If you take normal mammalian cells and you put them into the cell culture, you know, spare cell into the glass dish with the nutrients and the specific buffers, they start to divide. How much? Until they fill the glass surface, until they form one layer of cells. And then they stop. Why? There is no space, lack of nutrients, lack of oxygen, and lack of way to eliminate the waste. Cell feels the lack, that environment change. And that's called the density-dependent inhibition.
Is that why the medium is turning yellow?
I think medium is turning yellow because of the waste they're removing.
Okay.
Probably depending on the type of a cell too. So if you scrape some cells, how do we know this? Well, if you scrape some cells away and you make a gap, they're going to start dividing again until they fill the gap, and then they'll stop again. And these are the normal fibroblasts in a glass dish. Now, this is the problem. What we've realized is that transformed cells that we now call cancer cells don't stop dividing. They don't feel the density dependent inhibition. They keep dividing even if there is a lack of space, lack of nutrients, lack of oxygen. And they can divide indefinitely. What you need to know about the normal cells, epithelial cells, and other cells that have ability to divide is that they have a certain number of divisions they can go through. 20 to 40, I don't know, 60, and then they stop. So they have a certain number depending on telomerases, but we won't go into that. Now, what's interesting about the cancer cells, they have ability to evade that, and they can divide indefinitely. They are immortal cells, and we can keep. You heard about HeLa cells, right? And they've been kept in the culture how long, Peter? 50, 60 years? Since 50s, I think. There is a book about it now. So there is something about, now you have to understand one thing. We'll see that cancer cells are our cells that are transformed due to the certain factors. But what the main property of these cells is that they don't stop dividing for two reasons. First, because they don't feel the density-dependent inhibition, and the second, they don't have anchorage dependence. That's another thing that's characteristic for normal cells in the culture. You put them in a dish, and they can divide only if they anchor to the surface. Well, you can say, well, there is no surface in my body. Well, there is, right? All the cells are connected in a way, except maybe for, blood cells that are floating in the plasma. But all other cells are connected either to other cells or to the basement membrane. So they feel that connection. Now, they can divide only if they are attached, but cancer cells don't. So they can divide even if they don't attach to the surface. And that's another problem that we'll see how it manifests itself. So the main properties of these transformed cells is the unusual number of chromosomes. We'll see that underlying change in that transformed cell is actually the change in genetic material, the change in the chromosomes. That causes abnormal metabolism. There is loss of normal cellular functions, loss of density-dependent inhibition, loss of anchorage dependence. And actually, we'll see that they're totally independent. They don't care about anything. They're self-sufficient. They're selfish. And their only goal in life is to divide. So those are your cells that are losing the control over the division. So these are some epithelial skin tumor cells. Now, we can go back and just remind you about epithelial tissue. We said that epithelial tissue has the highest capacity for cell division. So no wonder that 90% of all cancers are the cancers of epithelial tissue, because those are the ones that have the ability to divide. Okay, so 90% of all cancers are actually cancers of epithelial tissue. So how can you recognize? So if you take a slide, a histology slide, you can recognize easily the tumors, the cancer cells, because there is a lot of cells in cell division, in cell mitosis. So you can often recognize some of those structures like mitotic spindle within the cell. And also there is a difference in the ratio between the nucleus and cytoplasm, because when the cell is very quickly dividing, it doesn't have time to actually replenish the size, to grow to the full size. So cytoplasms, the ratio between nucleus and cytoplasm is increasing. They have proportionally less and less cytoplasm, and more of it is a nucleus. Here is another fast-growing tumor that has a lot of, you see, a lot of cells in mitosis, and also the morphology of the nucleus is rapidly changing. Now, everything usually, in most cases, starts from one cell. Okay, one cell is changed. So I like to call it a transformed cell. It is going to eventually, maybe, grow into the cancer. I don't want to scare you, but you produce those transformed, changed cells all the time. So what's happening? Well, we usually have a very good control mechanisms that are either removing them, right, through the immune system, or they can sense that something is wrong with them and start the apoptosis or program suicide. So most of those cells are removed before they can multiply and form tumors. But those transformed cells happen all the time. Now, what is the cancer? What is the tumor? The tumor is when these cells evade all these control mechanisms and all these defense mechanisms and start growing on their own. So what is the process of growing? From the normal tissue, we have first metaplasia. So one cell, one of these cells, is transformed and starts dividing. And of course, the problem is they divide very quickly and don't care about anything else but dividing. And as they rapidly divide, they lose their normal appearance, they lose their normal morphology. We have a diversity in nuclear size and shape, and there is a dysplasia. So the first level is after metaplasia, or just change in appearance, we have dysplasia, which you have a part of the tissue that completely is changed its appearance and the function. Now, if this dysplasia continues, then it can form a neoplasia, which means the new growth. So what's now happening is these cells are growing over the size of its normal tissue and forming the clump of cells, neoplasia, or if you want to call it a tumor. That doesn't mean we still, well, there is, I guess, I'm glad Angela is not here, because there is, I think, disagreement between the scientists and clinicians. What do you call cancer? What do you call tumor? Usually cancer clinicians call the malignant tumors, but not every tumor. So here you have only the neoplasia or a tumor, but not necessarily the cancer. So we already said that cancers or tumors are the progressive pathologic proliferation of cells without the control. And there are two types, as you know. One is the benign tumor. It's just the growth. It's slow growing. It has usually the capsule. It's non-invasive, so it doesn't metastasize. And guess what? It's usually uncomfortable, but not life-threatening. What's life-threatening in, I don't know, 98% of the cases is malignant tumor. And every benign tumor could potentially turn into malignant. And there are some that have more chance to turn into malignant and some that have less chance to turn into malignant. But this is what you want to avoid, because this is what's life-threatening. This is what's causing the fatalities. And those are the fast-growing that are not capsulated, and they're invasive. So the main thing we'll see is what's happening with these malignant tumors is if the cell from a growth detaches and starts moving through blood or lymph system to the other parts of the body, invades other parts of the body, and starts growing invasively in other tissues. And this is what's causing the fatalities. Yes, detachment and invasion of other tissues. So in most cases, the fatality comes out of the secondary tumors, not the primary tumor. That means you can see all, you know, the neoplasia. You can see the malignant changes in all the cells. So it's, you know, you can identify easily the tumor in the tissue. Okay, and these are certain properties of a malignant transformation, or if you want, they're called the hallmark of cancer. And there is a famous review paper from 2000 called Hallmarks of Cancer that define these 7 properties of malignant tissue. And let's say just a few words about each of those. So the first one is that these cancer cells are self-sufficient. What does that mean? Well, I told you that normal cell responds to the outside and inside signals in order to start the cell division, the cell replication. And those are usually what we call the growth signals. Well, guess what? These cancer cells don't need growth signals. Or, even worse, they produce their own proteins that act on themselves as growth signals. So they say, okay, I don't want to play your game. I'm self-sufficient. I'm going to do it by myself. I don't depend on anybody else. And there are certain genes that are called oncogenes that promote this self-sufficient growth. So what are the oncogenes? Where are they coming from? You know, people were puzzled for a long time. What's happening? Why some people have oncogenes and the other people don't have oncogenes? Well, the oncogenes are actually nothing else but the normal genes in your body, certain genes that are in charge of the control of the growth and division of cells that are transformed by mutagenesis. So mutated genes that are in charge of growth and division are actually turning into what we call the oncogenes. So normal genes that we call proto-oncogenes are regulating the cell replication in the normal cell. But if they mutate, if they change, they turn into the oncogenes which promote the cell division in the absence of stimulation. And you can see that they produce special kinds of proteins called oncoproteins. So how are these oncogenes formed? Well, there are different mechanisms. And yes, it's true that I don't know, do I have slide, next slide? No, I don't. Oh, I have to go back. Some of these oncogenes are formed due to the viral modification. So yes, about 20% of all cancers are viral in origin. Why? Because the virus has ability to insert their own DNA into our genetic material and modify this signaling process. But only about 20% of these viruses can turn proto-oncogenes into the oncogenes. There are other mechanisms like amplification of genes, of course mutations, point mutations, deletions, or translocation of genetic material can all change these genes from proto-oncogenes into the oncogene. Now, what is the good news? The good news is, so we know that the cancer cells are starting as the normal cells that are genetically transformed because they have some kind of genetic change in the control mechanism, either some growth factor or growth inhibitor or some other protein that's in charge of cell division. But guess what? In most cases, I don't want to say all cases because in biology, everything, there is always exception. But in most cases, you need more than one change, change in order to produce cancer. Why? Because usually one change either is not sufficient or it can be dealt with by the body. Your body can see this is not good, I'll destroy myself, or my immune cell is going to eat that cell up and destroy it. So what you need is actually progressive genetic change in order to get to the malignant tumor. So there is some kind of multi-step process that causes the cancer. For example, this is a well-established model. It starts with the normal epithelial cells in a colon, and then there is first the loss of tumor suppressor gene, APC, causing a small benign polyps. That's why you do colonoscopy, to remove them. Because why? These small benign tumors can, by activation of some other oncogenes, cause the larger benign tumors, and especially this one is very important, P53. That's a suppressor gene or tumor suppressor protein. If that loses its normal function, you're in trouble. And that can cause the formation of malignant tumors. So there is a multi-step process, but you also heard that some people have more chance or a higher possibility of acquiring certain cancers, and they do genetic screening. Why? Because we know what genes need to get transformed in order to get certain cancers. And if you already carry one or maybe two genetic mutations or transformations that can lead to that cancer, of course your chance to get that cancer is higher. If you're starting here from zero and you need to acquire 5 different mutations in order to get colon cancer, yes, you can, but the possibility is much lower. But if in your family you already have mutations on 2 genes that are leading to colon cancer, you carry certain predisposition for cancer, then of course your ability to acquire during the lifetime the next 2 or 3 mutations is much higher. That's why if they say if you have a certain family history of cancers, it's a good idea to do the genetic screening for those genes. And of course, we now know more and more about that. The other property of malignant transformation is that the cancer cells are insensitive to growth inhibitory signals. And there are certain signals, and I told you that one important one is the signaling pathway for apoptosis, and that can sense the changes and abnormalities and start the destruction of the cell. But if these pathways suffer any kind of change or mutation and are not functional, there is no inhibition of growth. Especially P53 is very important for starting apoptosis. That's called tumor suppressor gene. And if nothing is suppressing the division of transformed cell, then of course they can turn into malignant very quickly. And these are just for your to understand the difference between the apoptosis and necrosis. Necrosis happens also all the time in your body, but it's very different from apoptosis. Apoptosis is, attentional destruction of the cell. Necrosis is something, it's an accidental cell death. It, of course, happens all the time if cell is injured or in any way it doesn't function well. But the way they lose their normal abilities and die is very, very different. Now, cancer cells can evade apoptosis. That's also very important. So, as I say, if you have a transformed cell, either apoptosis or immune system can take care of it. But cancer cells are smart. They have abilities to avoid the apoptosis by producing certain proteins that are protecting the cells from apoptosis. And especially mutations in this P53 gene are very important for that property of the cancer cell. There is another mechanism that's very active during the cell replication or division, specifically during the S phase, and that's the control repair mechanism. So you know that our DNA has millions of nucleotide pairs, and of course during the replication there are mistakes, but there is, and they happen all the time, but there is several sets of proteins that are checking as the DNA is replicated. They're checking the sequence and making repairs. And that's very important, of course, that the genetic message or information is transferred to the new cells correctly. However, if there is a problem, a mutation, a change in the repair mechanism, guess what? The DNA molecules, your genes, are going to suffer mutations. They're not going to be repaired. And it's very possible that some of these changes or mutations that are not repaired are going to be on the genes that are regulating the cell growth, the cell cycle, or they're inhibiting the growth signals. So these abnormalities of DNA repair genes allow mutations in the other genes that might have effect on developing of cancer. They also have limitless replicative potential. I told you that, yes?
I have 2 questions.
Right.
And what's the template for the repair the DNA?
The template for repairing.
Yeah, I mean, if you want to repair something, you need a standard.
The standard is depending on when it occurs. Okay, but in most cases, you know how the replication takes place. The DNA, the double helix, opens up, right? And each chain is the template for the new chain.
Okay.
And then, of course, in the new chain, there are possible mistakes. So the most often what's happening, if the mistake, and as the new chain is formed, the repair mechanism, which is usually a complex of proteins, is literally attaching to the new chains and checking it as they're formed.
Okay.
And if it finds the mistake, it cuts it off, insides that part, and then inserts the new. So basically, your old DNA is your reference for complementary chain.
Okay. I mean, it only happens when you like producing it cell by cell. And after it produced,
you mean the new DNA? Yeah, there are some mechanisms. Most mechanisms of repair occur during the replication itself.
Okay.
But there are some that are occurring afterwards as well, where the set of proteins attaches to the new DNA and just checks it. But usually your old complementary chain is used as a template to insert the correct nucleotides. Is that the two questions or one question? You said you have two questions. Okay. I told you that normal cells in your body have a certain limited number of replications, right? Depending on a type of the cell, but it's between 20 and 40, I think, replication, then it stops. But that's not the case with the cancer cells. And that has to do something with the mechanism of replication, because I don't want to go in details, but every time the DNA molecule is replicated, it can't start to replicate or duplicate itself from the very beginning. That part is called the telomere. It's made out of the useless, repetitive nucleotide sequences. Their only purpose is a placeholder. Okay, so every time the DNA replicates itself, the part of that DNA is lost. So guess what? With every each DNA replication, you have less and less of DNA chain on the very end. And of course, once you run out of those useless sequences, that's it. You can't replicate DNA anymore. Now, cancer cells. It's interesting that our own transformed cancer cells have ability that every time they replicate DNA, they add that useless part. They have ability to add another useless sequence so they can replicate indefinitely, because they never lose that very edge of DNA molecule. Okay, but they're smart. They're smarter than our normal cells, but they're not good. They're evil. So another thing that cancer cell can do that our normal cells cannot do is start angiogenesis. What is angiogenesis and why is it important? Well, if you start replicating cells indefinitely, guess what? They would die without one thing. They wouldn't have enough nutrients, they wouldn't have enough oxygen, and they wouldn't be able to remove the waste unless they're very close to a blood supply. Every and each cell in your body is at least 2 or 3 cells away from the nearest blood capillary in order to normally function. So the diffusion otherwise wouldn't work, wouldn't be efficient enough. So it has to be close enough to the blood capillary. So these cancer cells, you would think, okay, they'll grow and replicate and grow, and then what? They're gonna kill themselves because they're gonna starve themselves without oxygen and nutrients. Well, they're smarter than that. They have ability to stimulate the growth and development of their own blood vessels. So everywhere you have the growth of tumor, you have the growth of new blood vessels, and that's called angiogenesis.
And it's a lot of them.
And it's a lot of them. Because they replicate so quickly, they need a lot of nutrients and then they need a lot of oxygen. So they're not hungry and they have everything they need. So one of the treatments that's called the angiogenic switch. So one of the treatments of cancer especially fast developing cancer, is to actually attack the angiogenesis and stop the blood supply, and that way kill the tumor. And let's see what's next. Okay, this is probably the most important hallmark of cancer. Some scientists argue that other hallmarks of cancer can be found maybe in other tissues, but this one cannot. And that's the ability of metastasis and invasion. That's the main feature of malignancy. So this is the primary tumor in the breast. So the cells at certain point, and we don't know how and why, but we know one thing, that ECM or extracellular matrix have a very important function. Are detaching from the primary tumor. They have ability to squeeze in between the cells of the capillaries, get into the bloodstream, and travel all around your body, and then squeeze out of the capillaries, which other cells don't know how to do. No, well, some do. Sorry, macrophages. Some cells do, but most of the cells don't. And then start replicating or duplicating in other places. First, forming just the little groups called micrometastases, or we call them micromets. Or they can, if they start the angiogenesis, they can form the secondary tumors somewhere else. And guess what? Where do you think you can first indicate micromets? And that's one of the focuses of our, actually I'm involved in a project where we try to identify cancer early by looking, where would you look first? Lymph nodes, exactly. Because if the cancer cells are traveling through your body by blood system or lymph system, this is the first place where they're going to get stuck, possibly. So you look into the lymph nodes for micromets, especially if you have breast tumors. Okay. So this is the the example where cancer loses anchorage dependence and also density dependence, and just starts growing on its own and starts growing the new blood vessels. And of course, as I said, this is the major cause of morbidity and mortality. Yes?
So, first, the cancer started in the skin, it's a skin cancer, and it migrates and moves. Is it the skin cells that continue to reproduce in a different place, or do they cause the other tissue cells?
It's those tissues. Well, we don't know enough, but the cancer cell, the transformed cell, is the one that's actually replicating. But we now know, we're just starting to realize, that these cells that are invading other tissue make the new host cells work for them. Really. I mean, they enslave them. And not only did they work from them, but we now know that they have a potential ability to transform those cells. We don't know enough. But the general feeling is that, the general knowledge is that that cell that's moved and migrated and invaded the other tissue is the one that's going to divide and form the new tumor. But now we definitely know, and that's the new hallmark of cancer that you can find in, I can provide those references for you, they're very interesting. So there is like a brand new review from 2010 or 2011, the new hallmarks of cancer that actually lists 3 more hallmarks that were discovered in the past 10 years. And one of them is that actually the migrated cell that's invading the tissue is enslaving the healthy tissue and make it work for them. Yes?
So just to extend that off that question, so if you put a cancer cell inside someone's nerve system after an injury, could it potentially grow back nerves? Or?
No. The problem with that is that the cancer cells are totally de-differentiated. Your cancer cell, it was either epithelial cell or liver cell or some other cell, but once it's transformed, it totally de-differentiates. It doesn't do anything it's supposed to do as a skin cell or liver cell. It completely loses its morphology, anatomy, and function. The only thing it wants to do is to divide. So it's useless.
So you can never use it to reproduce damage.
No, for that you use the stem cells. Stem cells are dedifferentiated embryonic-like cells that can differentiate in any other cell, but still has ability to divide, high ability to divide. But unfortunately, cancer cells can't go back to differentiate in whatever cell you want. That's the problem. Okay. Any other questions? I think we need to wrap up. Others are waiting. So, formation of cancer, you have to remember it's a multi-step process at both genetic level and a phenotypic level, which means, phenotype, the appearance, the cell morphology and function. And the fundamental rules is that it's genetic damage, but non-lethal. Now, keep in mind that there are a lot of genetic damages and changes and mutations that are happening, but if they are not compatible with normal life of the cell dies and can't form cancer. So it has to be a genetic change, but non-lethal change. Change that won't actually cause cell to die immediately. It's a clonal proliferation of the cell, and the main hallmark is the ability of this cell to migrate and invade other tissues. And we are just about learning more and more about these cells and how important the extracellular environment or extracellular matrix is for migration and proliferation of cells. What we now know is that migration of these cells and invading the other tissue depends strongly on the consistency of matrix. For example, the stiffness and the content of the matrix. So some and extracellular environments are more prone for migration than the others. So very often, clinicians can predict if you have a primary tumor in a breast that it's going to invade specific tissue, not just every tissue in your body. It's going to, let's say, invade the nervous system and the liver, but not intestines and so on. Any other questions? Yes?
You said that our body normally produces these deformed cells, but
they happen.
So,
those are mistakes.
So do carcinogens, do they prevent the body from killing these cells, or do they actually cause the cells to become dead?
Cancerogens in our way of life are actually promoting or stimulating the percentage of those mistakes. And then the body doesn't have ability to deal with them. That's the problem. If you're stressed, if you're tired, if you don't have the normal life, if you don't eat the right foods, if you are exposed to certain cancerogens, you just increase the number of those mistakes, and then your body just can't catch up. And that's why, usually the number of cancers increases with age. Because everything just slows down and it's not working as well as when you're young, and just the number of mistakes and genetic mutations is accumulating. It's just about being able to deal with those. Anything else? Yes.
All these hallmarks of cancer.
Right.
There's so many of them, and it feels like these cells are optimized to do damage.
Yes.
So
they are very optimized. They're very clever.
So are these hallmarks actually from some common fundamental cause, or are they separate factors that kind of come together?
Well, that's a problem. They're coming from different factors, different. There is no one cause of cancer, but the problem is that they affect the same thing, and there's the control mechanisms for cell cycle and cell division. So different things can cause these mutations. They can happen spontaneously, if that's your question. And yes, they're all, you see, we believe that cancer evolved just like any other disease. In order to sustain, in order to stick around, it had to find the ways, to evade the apoptosis, to not need the space to divide, the ability to move through the capillaries in order to get into the bloodstreams, to migrate into the other tissues. So evolved. Very often they say that the cancer actually follows the same principles of Darwinian evolution like we do.
So you may evolve in like several years or several months?
Oh, you mean cancer in one person?
Yeah.
I think cancer evolved as a disease through evolution, especially with the human civilization. But yes, cancer can be very aggressive, or it can, have seeds early and then evolve over decades.
I mean, how can like cancer cell pass from one generation to another generation?
Oh no, you can't pass from one generation.
So
and just saying,
if it's evolution, it's only revolution inside your body, like through like several years until you die.
Right. It's acquired. It's not genetically. But I'm saying the disease itself.
Okay.
In order to sustain itself, just like you have the species that we don't know even about because they died because they were not adapted. The cancer is a disease had to adapt in different ways in order to continue.
Okay.
Is it possible that the study of cancer that the new additives and preservatives and fast food that
that's definitely true too.
That it caused a new kind of cancer and wasn't the old
possible.
cancer that modified?
We don't know about that. Yes.
So I'm kind of confused as to how cancer would evolve because as cancer tries to survive in a person, the ultimate result would be death.
Okay, how it evolves? Are you familiar with Darwinian evolution?
Yes, but
you have a lot of species, right? And some are adapted and survive and go on. Some don't adapt and they don't survive. So cancer also, as you can see, has different mechanisms, right? Very different mechanisms. We now have hallmarks, but it's not predictable. Are you going to have mutation here and there, and what kind of mutation you're going to have in order to get to cancer? There are different ways to get the cancer. Okay. Now the ways that are successful stayed, and we know about them. The ways that are not successful just didn't stay.
No, what I mean is like, for example, for cancer is starting to evolve. It will take the path that it's most likely to survive within the person. And then if it succeeds in surviving, then the person dies. How does the cancer pass on?
No, what I want to do, it's not about passing. It's about the fact that only the successful ways of developing cancer remain, and we can now identify. That there are potential ways of developing cancer that are just not successful and they didn't survive. We don't know about them. That's why they say that it has the same principles as the Darwinian, because there are multiple ways to get the cancer, many different ways, depending on the order of mutations, where they occur, on what the genes, in what sequence. Are they going to be killed, not going to be killed? So now we know what leads to the cancer, but there are many ways to get there.
Any studies going on to identify how to increase the strength of the immune system?
Of course, absolutely. There are a lot of different studies on angiogenesis, studies on following mutations, on immune system, and we don't know enough about what's the involvement of immune system in the fight of cancer. Okay. We know very little.
Do you know?
It's up to your generation. Come on. Yes.
So this is affect the whole body, right? So why it's not important, for the other organs to, because it blocks angiogenesis in the whole body, right?
What blocks the angiogenesis in the whole body?
The anti-angiogenesis drugs. You mentioned that.
Yeah, well, I don't know much about the science that's going behind angiogenesis, but we are talking now more and more about the targeted treatments, not the whole body treatments. We are trying to go away from the, holistic treatments and go to the, targeted treatment. To attack, for example, during the development, I don't want to go in details, but during the development of blood vessels, there are specific proteins that are found in those blood vessels, specifically integrins. So you can make special devices. Some of them are called microspheres that have, they're like made out of proteins and lipids, and they're little floating devices, if you want to call them that way, that you can target with the antibodies, specific antibodies, and you can load them with the drugs. And once injected, just like contrast agents, they go straight to the target. Of course, they affect other bodies, but other parts of the body, but less. So this is just one example. I'm giving you an example how you can do that. You can target the tumor, you can target the cancer or intensive angiogenesis with those microspheres and release the drug on the target.
So the receptor that make the angiogenesis in tumor is different than the one that...
Sometimes different, and sometimes there are what we call, I don't have time to talk about it, about biomarkers. For example, breast cancer, 30% of breast cancers have the special marker called HER2. And so you can devise the contrast agents or targeted drugs with antibody to HER2 that will bind only or for the most part to the breast cancer and try the targeted treatment. And we're going toward that because we think that's the solution, not only because you won't hurt the other parts of the body, but because your treatment would be much more efficient if you deliver the drug to the spot. So there are biomarkers. They're not as specific as we would like them to be. And yes, you can hurt the other parts of the body, but it's better than the holistic approach. Sorry, I think I see the signals to get off. Thank you, guys.